Okay, did you get a chance to look at any of the emails I sent yesterday?
Yeah, I was looking at some of them. You're talking about some. Well, first of all, you're talking about some of the ideas that you had for like different open questions and.
Yeah, and then I came across this paper on trees.
Yeah.
It seems to me they've rediscovered the differentiation tree, but I'm not absolutely sure. But I got a reply from one of the authors. I think I sent it to you this morning.
Yeah. Oh, yeah. I saw that email. So the paper is a reference tree. A reference cell tree will serve science better than a reference cell atlas.
Yeah. So I just put it as a question: is his reference tree the same thing as a differentiation tree, or I think it's vaguer. It's not.
Yeah.
Now I looked up the follow-up papers. There are only 3 papers that have referred to that paper, and none of them seem to get into trees.
Oh yeah. This was from 2023?
I don't know what was the date on his paper. I forgot.
It says 2023.
Oh, it might be.
Yeah. Okay. So, but yeah, they don't seem to. They say tree-based nomenclature, which doesn't mean tree.
Yeah, but we're trying to hang things on a tree too.
Right.
Anyway, read the paper and see what you think about similarities and differences. Okay?
Okay.
I think it might be something we should refer to, but I put a note there that maybe we can get him into axolotls.
Yeah.
Now, what we need actually is an axolotl that's transparent.
Okay. Yeah.
So you can all the cells.
Yeah.
Okay.
Oh, hi, Vahid.
Got here. Vahid,
how are you?
Hello.
Hello. How are you?
All right.
Tired.
Yeah.
Okay. So anyway, good to meet you. Vahid, you just starting a project or what?
Actually, I'm talking to Bradly for a while in the past. But, again, I'm rejoining the whole project, the open project again. I mainly work on channels, but now these days I'm trying to find some interaction between my previous works and what's going on at DevoWorm. I hope to be useful. Actually, I'm having some. I devoted some enough time these days for the whole project, so I'm trying to find a puzzle I could fit.
Yeah. He's been doing a lot of stuff with like Channel Worm and OpenWorm. Like OpenWorm has a number of projects, and they're doing things with ion channels, and he's done a lot of really good work with that. So.
Okay, so you got him pretty busy.
Yeah. Well, yeah. I mean, well, he's doing that, but like we talked before about like doing things in development too. So.
Okay, because I'm also looking for somebody who wants to work on a new form of computed tomography. Okay, which is very different from DevoWorm. This is aimed at detecting small tumors at low X-ray dose. Okay, so if you know somebody who might be interested, let me know. Yeah. I've got a kid working with me on the project right now. He's just out of high school, and he starts college in January. So starting January, I won't see him again. So he's doing very, very well. Chinese kid who lives nearby.
Yeah. So yeah, so I saw that paper, and then you sent some other papers alongside that.
Yeah, it looks like I don't know if I sent you the grantsmanship paper. Like maybe I did.
Which one was that? The Quake
I
paper or
called,
the?
I called one by Quake. Yeah, Quake.
Okay, yeah, that one. Yeah, I see it.
Yeah, yeah. This is a guy who's very proud of having raised $600 million.
Oh. for a cell atlas.
Yeah, they're trying to make this project as big as the human genetics project.
Yeah. This is the, which one is this?
Yeah, I think that's him. Quake, yeah, Quake. This is mostly about how good he is at raising money.
Right. They kind of go through their project and go through this.
Anyway, I think the trees thing is a good antidote to this paper.
Yeah.
It says, okay, where are you going to hang all this data?
Right.
And the problem is these people need lineage trees, and from our point of view, they need differentiation trees. Okay?
Yeah.
And getting a lineage tree from a mouse brain is pretty hard. It's opaque.
Right.
Okay?
Yeah.
You might be able to do it with a fish, like a zebrafish. You know, something that's a bit more transparent.
Right.
But I don't know. I think these people are they're knocking themselves. They're trying to make a big deal about making a list of how many cells there are.
Right.
And I don't know if I sent you what I wrote about it. The number of cells they find is about the same as what people knew 20 years ago.
Right.
But didn't make such a fuss about it. Okay. And the popular article I sent you ignores that work and goes back another 20 years to stuff that's wrong, or at least insufficient.
Which one is that? This one or the Tam and
No,
Ho?
it's the oh shoot, what do you call it? It was a popular article. Oh wait, go back to your list.
Oh.
Go back to your list. It's no, it's not there. Let me get to the reference. I'll put it in the chat.
Okay.
Oh boy. Oh, let me try another tactic here.
All right.
Here it is. It's from Quanta magazine.
Oh, okay.
Okay, by Saplakoglu. Saplakoglu.
All right.
And it was published this month. Yes, that's it.
Yeah.
Now, see, this one mentions cell counts for mammals as low as 500. Okay,
Yeah.
and that goes back to the 80s. The work that showed about 7,000 cells in mice, cell types in mouse, goes back to I think 1998.
Okay.
Okay, and now they've rediscovered about the same amount, and they don't give these guys any credit.
Oh.
That's the problem with being 80 years old. You know too much.
Yeah. Oh, this was Stephen Quake. They cite Stephen Quake in here, and
Yeah, that's where we got the quick. Yeah, this one is good for getting some key references. But when you get into it, you can see there's probably thousands of papers.
right.
Okay, so all these people are struggling with their cell atlases, which have no coordinates.
Right.
In other
Yeah.
words, they don't know where the cells are. They don't know the lineage, and they're trying desperately to figure that out because they know they need that.
Right.
Okay, and the trees and some other articles make that very clear. It took me a while to figure out that atlas does not mean atlas.
Yeah.
I mean, an atlas is supposed to be a map,
Right.
not just a list.
Yeah, a lot of the stuff they have is like they sometimes they'll put images up and they'll have like a stack of images and but they won't have any other information. It's just kind of like
that's why I got into it. I was trying to make sense of what's an S, what's a cell atlas,
right.
What is it, or a brain atlas? What is it? And it isn't. It isn't an atlas, but they call it that to get money.
Right.
Okay, so I think we will end up joining the chorus and goes to say, hold on, you're going to have to do your work and get your cell lineages and get real ones.
Yeah.
I sent you one article on a strange method which labels a whole bunch of beads, and then it takes an epithelial with DNA, and each one has a different DNA on it. Then they put an epithelial tissue on top of these beads, and this gets into the cells somehow, and then they map that.
Huh.
Well, it only works for epithelia, and I doubt it works well.
Yeah. I don't know if I got
that. Of course, it's very destructive, so I doubt they're getting lineages.
Right. I don't know if they got that one yet.
It's touted as the best method for getting the cell lineage. Yeah, there it is. Slide tags.
Slide tags. This, the single nucleus barcoding for multimodal spatial genomics.
Yeah, this is garbage. It's sort of how to look at an embryo through frosted glass.
Yeah. Huh. Yeah, I don't know.
Okay. So at any rate, I thought they had something, but it's clear that all they have is money.
Yeah. So I mean, it's interesting because there's been a lot of work done on like cell tracking, as we've seen in the meetings, and then there's been all this work on atlases, but people haven't necessarily brought them together.
I think the problem is they use animals that are opaque and have tiny cells, and the axolotl at least had big cells. It's still opaque. Some of the fish, I think, might provide transparency.
Yeah.
I don't know, maybe that's a place to search. There we go.
Like in what? In like zebrafish or in?
Well, zebrafish is the most popular fish. There's no question of that.
Yeah.
But, maybe Vahid can look up cell sizes in fish and find a fish with big cells. And then we see if it's transparent. Some of them have lots of pigment, some don't have pigment until later. I used to follow, it was a long time, probably 40 years ago, follow the literature on larval fish. Okay?
Yeah.
They're kind of interesting, but they're usually quite transparent at early stages.
Oh, okay.
Okay? So that's a possibility. I think what we need is a model organism for cell lineage tracking in vertebrates.
Yeah.
We got it already in nematodes. Now can we get one in vertebrates? Larval fish might be the best bet. If any of them have, especially if we can get it. One way to get large cells, by the way, is polyploid.
Yeah.
It works for axolotls. Axolotls, I think, have been done up to 7 ploidy. And you get 2 effects from this. The animal size is the same, the cells are much bigger, and they're stupid.
Yeah.
Because they don't have as many cells in their brain.
Right.
Okay. So if that can be, that was done in the 30s in axolotls, almost 100 years ago. So if you could do that for a transparent fish, you might be able to create a polyploid in which you could do lineage tracking. Okay.
Yeah.
So I don't know. Are you game for that, Vahid? See if you can find any literature on cell sizes in fish.
Actually, as I said, I'm not yet delved into the lineage of actually something that it's been happening right now in the DevoWorm. But we have done, and I know students over here that are working on the mechanical aspects of the movement of fish. So if there could be any maybe for the somehow for the simulation of the movement, if there could be any after of course we have done some work on the lineage part, and we could somehow step by step go from the small size fish to when it grows up, we might be able to from my side, I could maybe do something for the.
Well, you don't need a big fish. The larval
fish.
For
sure. Yeah, especially.
Larval fish are small enough. I wrote a paper once on putting larval zebrafish in a capillary tube, and this would allow one to see the fish. You could rotate the tube and see the fish at any angle. Okay, so and then you could do computer tomography and do 3D structure. So it's not out of the question. I think the trick is to find there are many species of fish. Let's see. I'm going to look that up actually. I'll look it up. Number of fish species. There we go. How many species? 33,000 known. Okay, 33,000 known species of fish. So amongst those, there might be one with big cells.
So, send me, I think, a few more papers.
I read them and then I send them to you. But
yeah,
I've got about 80, 90 papers.
Oh wow! Oh, that was the list. You sent me the reference list. Yeah. So this is. I think this is just 9 that you sent me, but I don't know. Yeah.
Oh, okay. This is my bibliography, and it's this is the list. Okay. Let's see, so you can see more of it. Now, the paperclip means I have a copy.
Oh, okay.
You see, I have a lot of work to do in getting copies.
Yeah.
Okay. But this is what I've got so far. By picking out review articles, hot articles, etc.
Yeah.
Okay. Like this one, for instance. Single cell lineage tracing.
Yeah. It's like I see cell atlas, I see cell tracking, spatial like spatial omics
Yeah,
stuff.
I mean, it's clear that
yeah.
this field, they're hoping for a breakthrough, but they need to be able to track cells.
Yeah, that's great. Should be pretty interesting to see all the references there. Yeah, you had some other papers in the thing that you sent me. This one was cellular diversity and lineage trajectory. This was from mouse single cell transcriptomics. So you had a couple of things outlined here.
I've been trying to read these. Well, I started reading these papers carefully until I understood that an atlas is not an atlas.
Yeah.
Some of them hope to get a lineage tree someday.
Right.
And but nobody's doing it right.
Right. So
this one, Patrick Tam. I wrote to him last night. I haven't had a reply yet.
Yeah.
I don't know if you recall, I did that work on salamander neural plate with Anton Jacobson back in the 70s or so. Anyway, Patrick Tam was a postdoc with Jacobson, so that's how I knew him.
Oh, okay. And so yeah, these atlases allow the dissection, not build, of developmental cell lineages and molecular changes during embryogenesis. And then advances in computational techniques have enabled the inference of developmental lineages in differentiating cells, even without explicitly introduced genetic markers, meaning you can identify them from like this different gene expression patterns or whatever. And then they talk about this, and then they have this passage here. It is evident that bulk transcriptome analysis only provide an average of genome activity. So this is the average, so you would get like a bunch of some signature, but that's only an average because they do not take the heterogeneity of cell populations into account. The average modality, go ahead.
By the way, the field is in real disarray, which they don't seem to recognize. Let me show you what I mean by that.
All right.
This is, I wrote a little bit more last night. This probably should not be a separate paper. But at any rate, here, look at this. Let's see, let me get this screen share back on.
Yeah.
Touch screen sharing. No. Okay. Here, look at this. Let us assume that the human brain is constructed using a simple bifurcating differentiation tree. The number of cell types is presently estimated as between 3,300 and 5,100. This is where I added the historical stuff. Roughly consistent with the 7,000 cell types found in whole mice. That was the 1998 with a total of 86 billion cells. Now look at this. Now that's for you.
Yeah.
How many times bigger is the human brain than the mouse brain?
Probably about maybe 2 or 3-fold. I don't know, depending on the structure. Yeah.
Well, here's a paper I should in mice. Okay, I found one of those papers discussing mice, and they find 140 million cells in the brain of a mouse.
Huh.
Okay.
Yeah.
So then I took the human data, if you want to call it data. That's 17 million to 26 million cells on average. That's the 3,351, and with the 86 billion estimate. Okay. So they're between 17 million. In other words, for each cell type, the average is somewhere between 17 and 26 million.
Right.
So I took powers of 2 for a very simple-minded differentiation tree. Okay?
Yeah.
And you get 2 to the 20. So these numbers fall between 2 to the 24th and 2 to the 25th, which means the portion of the, if there's a differentiation tree for the brain, it would have about 24, it would branch about 24, 25 times.
Yeah.
Okay. Nobody did calculations like this so far.
So this
anyway,
would,
I guess it should actually get the mass of the human brain and the mass of the mouse brain and just take the ratio, see if it's consistent.
yeah. Well, yeah, the cell,
I know.
yeah, the cell estimates aren't like they just kind of estimate from, I guess, tissue. I don't know how they actually make the estimates. Yeah.
I don't know. That's a lot of cells to deal with. But,
Yeah.
they're tackling it. So, I think there could be a breakthrough, but the breakthrough might be in finding the right animal.
Right.
With big cells and transparent brain.
Yeah.
Okay. So I suspect such an animal may exist.
Yeah. Well, the thing about like mammalian brains is that you have different structures, and they have different so in mammals, the neocortex has expanded greatly in different lineages.
Oh yeah, I know.
So
and some of the papers try to make estimates of those things.
Yeah.
Too.
But I mean, my point being is that you'd have a very different differentiation tree, you'd have maybe
I'd suspect though that the base of the tree would be similar.
Yeah, it would be probably.
Okay, so it's an interesting problem, and these people want to tackle it, but I don't think they're doing it right.
Yeah.
But I'd take a small portion of their 600 million.
Yeah.
I don't know. Zuckerberg, it's money he got from Zuckerberg.
Yeah.
And Zuckerberg is building a huge couple of $100 million bunker in Hawaii.
Oh, yeah. Yeah, they've funded a lot of the biomedical. They have a huge fund for biomedical research. The Chan Zuckerberg, whatever it is, but yeah, they definitely favor sort of the omics research. I think in a lot of it. So,
okay.
But I don't know. Yes, I mean this is again. Yeah, I get the sense that the literature is fragmented or the field is in disarray. It's just not. Doesn't seem like they're connecting everything together the way that you would need to.
No, and then I don't know. This tree paper seems to me is being ignored.
Yeah.
At any rate, I was surprised that one of the authors replied. The other one was probably a student because the email bounced.
Oh yeah.
Actually, regarding the fish you were talking about, I've been searching in my somehow bookmarks, and I'm somehow interested, as I said, in some topics like intersection between what I've done and models like ion channels. They related diseases like channelopathies, like Parkinson, etc. And what we could do with models like C. elegans or zebrafish. I found a paper, a work that has been done using zebrafish as the tool, but they have somehow been looking for a gene which is we could say common. They have some similar roots in zebra, but they could be useful for looking how Parkinson could actually be treated in children. So they are working basically and maybe from a lineage point of view, they have been worked on this disease from we can say how the genes in children are causing the Parkinson. There's some rare type of Parkinson which is really in some actually some children can have that type of Parkinson as a
Oh,
child,
really?
and
Okay.
yeah, and so if we are trying to look into these kind of applications, maybe I'm saying from my point of view, it would be really interesting. I could join you and work on some type of genes that are could be as a tool, could be studied in C. elegans or other type of animals when they are at the beginning of their growth or their
Okay.
different steps of life, and then actually we could see how we can use that type of tool for studying such diseases. This is one example, particular example. I would like to,
Yeah.
if it could be possible,
I didn't know.
join you.
I once had a bird that seemed to have Parkinson's at a very early stage.
There are
when
some
I was-
problems.
When I was a kid, but I thought Parkinson's was a disease mostly of old people.
Yeah, this is true, but it is a rare type of Parkinson, which is has some roots in genetics. Some type of children could
Okay. I didn't know,
have
I
that
didn't know
type of
Interesting. Okay.
due to some mutations, yeah. And so, as I said, it could be studied. Some similar diseases could be studied if we could look into some similar genes in C. elegans from the earlier stage of C. elegans, and also as we can see in zebrafish as well. If you have any similar issue or topic in mind, I would be really interested in jumping.
Is there any way you could use such genes for lineage tracking? Maybe. Okay. Because it seems that lineage tracking in a vertebrate is very difficult. Bradley, has anyone succeeded? Did you know?
Not really. I mean, I've seen a couple of papers. I don't know if we did them in the group where they talk about mouse. They were trying to reconstruct mouse lineage trees, but because the cells are not deterministic in terms of their fate. In other words,
Right.
like they're a product of signaling. They tried to infer the lineage trees instead, or they tried to like compute what they should look like, or yeah. So they tried to pick pairs. Yeah, I wish I could dig up that paper. I can't quite remember the citation, but they would generate like a number of candidates. So it was a little bit different approach. It would generate some candidates and then figure out which ones were most plausible, and then
I think the essential thing here is to have a transparent enough brain and a marker that could be tracked, in a lineage.
Right.
And to have big enough cells so it makes it easier. I don't know, maybe an Ascidian.
Maybe, yeah.
They have, because they have tadpoles, which they're basically the first vertebrates.
Yeah.
Okay, but I don't know about cell sizing. Okay, give me a second. I'm going to look up polyploidy.
All right. It's like Morgan joined us. Hello, Morgan. And Lucas.
Hey.
How are you?
Okay. Okay, this one's more modest. 39 papers I found. I've got.
Or polyploidy?
Yeah, I'm polyploid.
Oh, okay. Just polyploidy or polyploidy plus something else?
Well, just polyploid.
Oh, okay.
Okay. And the question is whether there's anything useful here, I don't know.
Is it mostly in plants or in like?
No, animals, mostly animals. But you can also do archaea. Okay. I think Jitsi is slowing my computer down here.
Oh. So yeah, just to finish up on this paper that we had, they talked about the averaging of bulk transcriptome, and so you don't necessarily know. And this is one of the things about the paper we were doing that we're revising right now. That we can get data from NCBI, but those are like averaged sort of transcriptome analyses or proteome analyses. They give you some insight into what's going on in a cell or in an organism, but they don't give you single cell resolution. Sometimes you can get single cell resolution, but it's hard to interpret that data in context. So there are a number of problems with like a lot of these sort of omics analyses and plugging them into a tree and getting data. So yeah, this has a nice image of human development from the blastocyst to the fetus, just showing the complexity of development. Says they talked about this paper, Pidgeon, Sala et al. 2019. They identified cell clusters representing the ectoderm, mesoderm, and endoderm lineages. So these are the three germ layers. And then the number of cells in each cell cluster progressively increases and diverges into subclusters as the embryo develops. So that's what we're dealing with. We have these three germ layers, and we have things that are diverging and increasing in number, but diverging into clusters. So that's something that we have to kind of think about. We think about that in terms of a tree, but they don't mention tree here really. It's just kind of like this is what's going on, and then they kind of give some more. This is in other studies, 38 to 98 cell clusters are identified in the mouse cell atlas at the organogenesis stage. Although most cell types expand in numbers exponentially, the abundance of cells of different clusters may appear proportionally relative to one another. So the different clusters sort of stay in proportion, and this is how you get different structures. So you get larger structures, smaller structures, but they kind of divide in their own sort of sublineage. And again, they don't use the word tree or lineage. They just kind of talk about clusters and precursors and things like that. So it's interesting how that language even is disconnected when they talk about these things.
Okay, the paper's on the polyploids on its way.
Okay, that's good. Any really good ones that stand out, or?
Well, I didn't get to read it. I just sent it to you.
All right, I'll take a look later.
Okay. Okay, but yeah, I think the polyploids are key to cell lineage. Okay.
Yeah.
Let's see some of those papers.
So let's see. Oh, there is this other article that you sent me. I don't know what this was. Is this like a letter? You sent this, the brain cell atlas, lineage trees, and differentiation trees.
Oh yeah, it's one I read last night.
Oh, okay. And this is the one that, so this is
the list I sent you goes back to 1949.
Oh wow, okay.
Okay, on polyploids.
All right.
The way you make, by the way you make a polyploid is you let the eggs start dividing, and I think they heat it up or something like that, and it prevents division of the cell while the nucleus divides. Okay, so you end up with a polyploid.
Yeah, they just kind of bunch up in the same cell.
Yeah. So it's an old technique, but it worked.
Yeah. Wonder if they have newer techniques for that.
I don't know. I don't think there's any interest in that work.
Oh.
Anyway, yeah, there are some more recent papers in that list. So
yeah,
I was just looking for the oldest one. Okay, what else you want to do?
Well, I guess that's pretty much what we had. Let's see if we have we talked about this one. The Stahl paper, I don't think we talked. This is the one that was the sort of the News and Views article about this other paper that you.
Oh yeah,
the slide tags paper, yeah.
Making it a breakthrough.
Yeah.
Yeah, well, that's the role of every science reporter.
Right.
Any research they report on must be a breakthrough.
Oh yeah. There was another thing that I think it was Morgan posted in one of the Slack channels, and this was a new atlas. So I'll bring it up now. Yeah. So this is a tridimensional atlas of the developing human head. So again, this is another atlas, but it's basically a similar thing where they basically take human embryos, they do tissue clearing. So they do, they clarify the tissue.
Is that still legal?
I don't know what stage they're at, but they're doing, yeah.
Well, no, I said, is it still legal?
I don't know.
We went through a period where you couldn't do anything with a human embryo.
Oh, yeah, I don't know. I guess it is because it's published in Cell. So, anyways, they did this tissue clearance, which will make tissues like transparent, but it's a process that doesn't allow you to recover anything. Like it would have to be extant. So, or it couldn't be extant. You wouldn't be able to get any more cell divisions. And then you have light sheet microscopy, so you acquire image stacks. And again, the image stacks, you can reference them spatially, but you still have these whatever is in the frame of the image, that's what you get. You have to then reconstruct it. Then they have this mesh reconstruction where they reconstruct inside they kind of infer from the image stack what I guess this is the nerves or connections between the cells. And then they do VR segmentation, which I guess they're using virtual reality to segment some of these images. Then they have a data portal, interactive website, and then the 3D printed model, which is where they print out the images. Yeah.
Okay.
So this is
Okay.
pretty, bringing all tools to the party, I guess.
Yeah, and it's very low resolution compared to 86 billion cells.
Yeah. So they're able to get this, head embryogenesis encompassing muscles, vasculature, cartilage, peripheral nerves, and exocrine glands. And then this data is used to provide insights into the branching morphogenesis of exocrine glands. So just one of those, the branching morphogenesis, they just want to look at what that network looks like. And then features of neurovascular and skeletal musculature structure development. So this really kind of does. So this is at Hough.com. Yeah.
Did they do it on twins?
Oh, I don't know. Let's look at the methods.
Probably not.
No, I don't think so.
It'd be hard to get twin embryos because then you could look at the variation.
Yeah. So, okay, a total of 76 specimens were used, including 27 embryos and 49 fetuses.
No twins.
No. And so they had male and female and 7 unknown. All images are from single samples and are not average reconstructions of multiple samples. So I don't know how that worked. I guess they stitched together. They just kind of glossed over the variation. I don't know.
Oh no, it said from single samples.
Oh, okay. So, yeah. The correspondence between samples and figure panels or movies is provided in table D1 or S1D, which is supplemental data here. I can't access it right now, but this is at houdeca.genos.org. This is the French Houdeca Biobank. So this is the actual website that they have, the atlas itself. So this was a 3-year program to build a reference of embryonic and fetal human cell types. So again, this is kind of what this. Yeah, so they have this, and then you can browse the omics data and a 3D collection. So, and then
Yeah, it's just awesome.
Yeah. And so this is an analysis of the chondrocranium development. So they have this is where they're looking at bone structure and kind of breaking down different parts of it. This is more, just anatomical. This isn't cell lineage, but it kind of gives you all this information here.
Yeah, so it's super useful for people who are looking at lifespan EEG, though, right?
Yeah.
When if you're getting EEG at different stages of development, and this is just taking it down to a level I never thought we would actually have.
Yeah.
But fetal imaging is also even in utero fetal imaging is really hot and just in imaging in general. RSNA kind of meetings. A lot of interest in fetal imaging, and so this kind of ground truth model is just great.
So why the interest in fetal imaging as opposed to other types of imaging?
Well, just an example in terms of this case, but again, it's just about getting the development right. So just like the organoids are starting to show kind of physiological rhythms at such early stages. Being able to capture the rhythms as they change, it's really interesting how sleep changes in the first years of life, right? So just your sleep EEG changes dramatically, or continuous recordings change so much over time. in the early years. Like being able to take that even further back would be, really valuable. And so getting that kind of data from, say, ICUs and is something that there's some groups here at UCSF that do that, right? It's only like single recording or single channel, maybe 2-channel, but I've talked to some of the people like trying to get them interested in a 64-channel system to one of these nets, but again, it's these are babies that are it's I don't want to say sick, but it's getting permission to work with them is very difficult when they're so fragile, so there's just by doing in vitro, in utero imaging and being able to do, say, MEG is something that people are interested in, just to get that kind of, just to see what that kind of change is like in earlier stages. And, tell us more. It's just looking at Slack while you guys were talking and saw Jesse's comments of, well, it was really, posting somebody else's Twitter, right?
Yeah.
About how, with entropy and complexity, we're in the age of measures looking for measures. And I was going to say something like, well, except the people who are doing neural modeling. Sorry, my dogs are fighting. But the neural modeling people are, with real generative models, you can hopefully say something actually physiological, right?
Yeah.
And just how the physiology is changing, is one of the easiest non-invasive ways to capture how the physiology is changing is from capturing that endogenous electrical activity, right?
Right.
So that's certainly the interest. But I would say also,
Morgan, what's the spatial resolution of that technique?
If like in utero imaging.
Well, in any such, yeah.
Yeah, so it depends. I mean, obviously, it's going to depend on the age of the fetus. Like the more the later the better in terms of the signal that you have, so I need to check on my 2 dogs. Sorry.
You can eat that bird.
So the one who's not letting the other into a bed.
Oh.
So when we have if the fetus is close to the belly where we got the doer. I mean, I'm talking MEG here. Although honestly, I don't think it would really matter. EEG would be. This isn't a case where MEG, I think, has a. I think the real benefit you get with the MEG is just you get the sensors in place super fast, but you probably don't get they probably don't get around the belly very well. The doers. I wonder what you could do with maybe OPMs now that allow you to have more flexibility on where the magnetic sensors can be placed. But anyway, we would say 5 to 10 millimeters, probably, which again is huge, right? So you're really going for kind of one single virtual channel. You're kind of using all your sensors to try and get one channel of data. how much how much is missing from that voxel, or how much extra stuff you've got in that voxel. It's a big noise. I mean, a big noise problem.
Okay.
Yeah.
Morgan, let me ask you a question. Are you familiar with electrical impedance tomography?
Yes, I'm actually one of the few people who probably is. Yeah. So we were doing it. We were doing it back in the '90s because we were trying to make an EEG system that could compete with an MEG system, right? And the MEG was able to, basically wasn't influenced by the skull and it's the spatial smoothing that the skull does with the EEG. So the idea that our CEO had, or the company that I joined had, was, we'll do electrical impedance tomography with a high density EEG net, and that would allow us to then use computational physics to see through the skull. But I can tell you about
I once designed an electrical impedance tomography device that would be used for embryos. Which might be one might be able to do it at very high resolution.
Yeah. Well, it was the paper that Amanda dropped in orthogonal Slack. I didn't recognize the author. It's an Asian name. I don't know if you know him, Bradley, but said that he was working in bioelectricity of embryogenesis.
Okay. I don't know. I haven't looked at it yet.
I think it's in like methods cognition. Sorry, let me just see. But methods cog sci projects. But that sounds really interesting, Dick. I'd love to. I mean, again, like this.
Okay. I helped supervise a PhD thesis on EIT for breast.
Yeah.
That's how I got interested.
So, I mean, I was going to EIT conferences back in like 2004, 2003, and
this would have been 20 years earlier.
Got it. Okay. Gotcha. Well, but that was definitely one of the application areas. Like us showing up doing the heads was, we were by ourselves.
I'd like to see, Bradley, that EIT could possibly solve the transparency problem for embryos.
Oh yeah.
Okay, because it'll go right through it doesn't care about pigmentation.
Yeah.
It's interesting, right? But it's also really interesting in terms of like your stuff and like you were the one that first told me that bioelectricity is important in embryogenesis.
It is, and unfortunately, my brethren will not talk to anybody.
Yeah. I mean, I was gonna say this is all 11 stuff, but
no, it isn't. It goes back 100 years.
Yeah, that's awesome. As someone who had to read Dreich in college, I think that's awesome.
Well, he didn't do electrical properties, but there. I think in a book I wrote, I think I listed this was published in '99, and I listed the papers that I found on electrical properties of fish embryos.
Interesting. Okay. Yeah.
Okay, which went back to around 1900.
Yeah. I mean, I'm looking for more references on this, and I would love anything you've got, you definitely recommend.
I haven't thought about it for years, but
well, I mean, is this a widely studied area now?
I haven't checked. I don't know.
Yeah. I mean, I'm super
I
curious.
doubt it. No, I doubt it. I think what I'm suggesting is that it might be practical to build an EIT.
Oh yeah. Yeah. I mean,
Microscope.
not only that, but no, what's her name? Mind's Eye Biomedical Imaging. Gene Rintoul and Gene's this awesome biomedical engineer who also presents at Chaos Computing, which is it's a big hacker convention. So you can see one of her talks at CCC, but she made a great kit for people that allows you to do EIT in a dish. And I don't think she's got a current run. Well, she did this, but then she started her PhD at Imperial, doing she never really told me what, but I guessed it was like photoacoustic imaging. Anyway, she started her PhD, and like 2 months later, pandemic lockdown happened. And she also had a new baby. And she had a hard time with that. But
that depends on the husband.
well, they were just trapped in a very small apartment in London. Is why it was hard. But yeah, definitely. But I'm super curious to know what she's working on now. But like her kit is, again, because she open sources all her designs and things, is just it's a perfect example of what you would make. I mean, if not, I mean, I don't know how small the embryos that you want to image are.
Probably half to a couple millimeters.
Okay, so yeah, so this is it would be shrinking her stuff down. I mean, getting it such that it's almost a chip these days is actually something that's pretty doable too. And the kind of equipment that they've been building to do what I would call more sophisticated cerebral organoid work is would be very very appropriate for it. I mean, Johns Hopkins built little mini helmets. I mean, they called them mini helmets. All they just meant was that because of the way you make the organoids, you end up with spheres. That it's appropriate. It's not, again, it's just it's good to say that it's not a brain.
Let me take a minute here, and I can. Let's see. There's a whiteboard on here. Let's see.
I'm gonna be getting some kids to school. So,
Okay.
but I'm listening.
Oh, okay.
I'm listening.
Okay. Can you see the whiteboard?
Yeah.
No, I can't see it yet. Or
okay. Let me. Where's the commands on this thing? Oh, there it is. Line. Okay, I'm just going to draw lines. Okay, so here is a baseboard, and here are electrodes. Okay, I won't draw them all. And here's an embryo.
I'm still not sharing.
Still not sharing. No, why not?
I can share my screen. That should work.
Can you see it, Bradley?
I can, yeah.
Oh, you can.
So there's my screen, if you can see it.
Okay, so
I can see that.
I started to draw an embryo, but it should be roughly a sphere sitting on top of these electrodes. Now, Morgan, am I correct that it's possible to address any pair of electrodes?
Well, yeah, I mean, or certainly that in our system, yeah.
Yeah, okay, so you address a pair of electrodes, the current goes out one and it goes through the embryo and comes back through the other. And that gives you one image, so to speak, of the embryo. And you do it for different pairs, and you build up the three-dimensional structure. Okay, so it's a very simple design. Obviously, hard to build, but and you know, if you use something like axolotls or fish embryos, you do it underwater. You could have the solution outside the embryo. Could usually be a weak saline solution. So you get conductivity through the solution too. Do you understand that? In other words,
yeah, I'm just sorry, a little busy here.
Okay. Okay, so you have a huge array of electrodes. You take any pair of them. You run current from one through that pair. It goes up into the embryo and down. That gives you a sort of electrical impedance image of that embryo through that pair. And then you change pairs. So you can get many views of an embryo, and it should be possible to calculate the 3D structure. As I said, it doesn't matter if the embryo is opaque or not. Okay, maybe we should do a paper more sophisticated than this on how to build an EIT microscope.
Yeah, it'd be interesting.
I think that would be awesome. I mean, again, I'll when I'm free later today, I will pull the equipment design that I forget if it was JHU or sometimes they outsource to the applied physics lab there, and
okay.
But anyway, like this should be. I mean, if Levin and the others that he's building on top of are correct, this is like really important to capture the bioelectric organization.
Yeah. It comes back. I mean, for instance, I don't know. Are you familiar with Lionel Jaffe's work on this?
Now that name does sound familiar. Why is that?
He worked on all kinds of waves in embryos.
Okay, then maybe it was something that because I read your paper. You had draft paper back a year ago now. Has that been published?
I don't know. I don't keep track.
Well, it probably is from one of your papers that I've seen that name before. Yeah.
Okay. Well, what Lionel did is he found that the neural plate of a chicken has a huge electric current that comes out of it.
Yes. Yeah. No, it was definitely you who told me this. Yeah.
So, and I don't think anyone did anything with it.
Wow, that's amazing that nobody's done it.
Yeah, well, new discoveries are beyond most people.
Well, like you said, it would be really important to have this kind of equipment. And
stupid question: can you use EIT equipment to measure active electricity in an embryo?
No, you go back and forth, right? So, I mean, it's you can do what we call it from this is the MRI term, but transmit and receive. So you do you use the high density to source image, and then the EIT to probe the material or probe the field.
Okay, but you can't use the same electrodes to pick up an electric signal generated by the embryo?
Well, you use the other electrodes, right? So I mean, this is the benefit of having, or certainly for us, the benefit of having like 256 channels is that we're doing random pairs to.
Oh, I'm thinking of more by five. It's 512 by 512.
Sure.
It's a two-dimensional array of electrodes.
Yeah. So you're using some to transmit and some to record. And
yes, if you can address which is which, yes.
Yeah. So that's all. Like again, like Gene's equipment, which is just a 32 channel, demo, like for 2D. It's cool for the kind of stuff that you do, kind of like MRI microscopy or diffusion weighted imaging. Take a slice of like a melon or something like that, right? And put it in this dish, and you can see you can capture some of the changes in structure. So yeah, you all of that's implemented in the microcontroller, however you made the electronics, but it's very doable, and again, like Levin says, he's stealing from neuroscience, stealing tools from neuroscience to study, bioelectricity's role in spatial morphogenesis. I mean, this would be a great one, because it was really great to see, what was her name, Ellen Kuhl, it's like Kuhl, the Living Matter Lab at Stanford, because they're doing all the kind of finite element modeling, and they were only familiar with things like MRI, elastography, and things like that. But I've wanted to get in touch with them, and tell them about electrical impedance tomography and other techniques that they could be using too. Yeah.
Okay. Well,
So the Living Matter Lab has as a focus brain tissue. It's like physics of brain, physics modeling of brain tissue. It's a rainy day here in San Francisco, and for some reason, kids are not getting up.
okay, go get them up.
I will be listening, but I'll be a little distracted here.
Okay, well, interesting. Maybe we can do something with this. Okay, Bradley, we may have solved the problem.
Okay. That's great.
Okay.
Yeah.
Because if electrical impedance tomography will work, as I said, it's independent of pigment. It'll go right through the embryo.
Okay.
And the question is, can we achieve the spatial resolution necessary through lineage trees?
Yeah, that's tougher, but
it's not that tough with nanotechnology nowadays.
Well, there's some limitations though in the kind of the far field measurements. So you're doing an inverse problem that
But far field for us is a millimeter or two.
Yeah. Now, what's cool though, and this is what I'm hoping to hear from Gene, is what people are trying to do in terms of combining like ultrasounds with electrical impedance tomography.
Oh, what?
And
yeah. What's the limiting spatial resolution on ultrasound?
Well, it's better, but even together, you can use physics approach and actually get, they complement each other. So you're able to, again, like let me let me get this this sleepyhead out, and I will follow up with some references and things that that I think would be really exciting.
Yeah, of course. Yeah. Anything else like X-ray? Well, you can do X-ray microscopical tomography now.
Yeah.
And,
yeah.
You know, so
it's hard to compete, and it's like people like, especially in like the focused ultrasound, researchers, they're typically using like microscopy or certainly optical imaging in a small scale animal prep that they can then, which kind of gives them ground truth, and then they can compare that against what they're getting with their focused ultrasound techniques and things like that. So it's hard to beat that.
When you get a chance, Bradley, you ever get recording the session, right?
Yeah.
Okay, when you get a chance, Morgan, you might want to review this session, because earlier we were discussing the problem of creating lineage trees for mammalian embryos. Okay.
It's I'd be curious. It'd be good to see what they are capable of.
They aren't yet. That's the problem.
But like you said, or as Levin shows, that like capturing the electric fields is a necessary, is a totally necessary, component of this, and so I think it'll be definitely a really interesting.
Oh yeah, I agree. I've been after Mike Levin for years to try to combine his bioelectric work with the differentiation tree approach, and he's just off in his own world. He won't budge.
He's off in a great world.
Oh yeah, but it's got its limitations.
Sure.
I mean, it's like looking at the moon and saying, "Gee, I see the moon can glow," and saying, "Well, what do you know about the geology of the moon?" Not much.
Yeah.
Little surface topology, maybe. But you got to go there and figure it out.
All right. Well, that's a great conversation. Yeah. Let's follow up on that and see what happens. Yeah.
Okay. Yeah, I think, with Morgan having the experience in EIT, this could be made quite realistic.
Yeah.
And it might solve the electrical impedance. It might solve the transparency problem.
Right.
Okay. Yeah. But I think that may be the key.
It's cool too, because there's packages that are like supported software packages available, like SimNibs, that weren't designed for kind of EIT. They were designed for transcranial electrical stimulation, kind of neuromodulation work. But they can totally, it's all there for you to use them as EIT modeling packages.
Oh, okay.
It's all good, finite element stuff.
Okay.
Again, I'm really interested to see what the Living Matter Lab thinks of this, and kind of, I'll say that, I mean, again, like I'm focused on the head because our subjects have been older.
Good. See, we've been looking at what are called cell atlases and brain atlases, and I concluded they are not atlases at all. Okay, but the same problem. They have not solved the problem of good lineage tracking or differentiation tree tracking cells in the brain.
Yeah.
Okay.
Yeah.
Uh,
Isn't it because they're destructive? I mean,
no,
Or is
it's
it
not because they're destructive. It's because they, in the living organism, they just can't get the resolution and to follow individual cells. So we've been talking about, okay, how about dealing with an organism with bigger cells? And we're talking about dealing with one that's transparent, but maybe with EIT and X-ray techniques, you don't need transparency either. Okay, so it might be possible.
Interesting. Yeah.
Okay.
Right.
Yeah, these people are struggling. They've got lots of money and they don't really know what to do with it because they would like to lead the next thing analogous to the Human Genome Project, but they don't know where to go. They're hoping for a breakthrough in the next 2 years.
Yeah, sounds great.
We've been going over that literature. So.
Yeah.
Okay, you want to call it a day?
Yeah, let's call it a day. Thanks for attending, everyone. And
Okay,
yeah,
take care.
take care.
Thank you.
Thank you.
Okay, bye.
Bye.
thank you, everyone.
Yeah, thank you.
Bye. Thank you. Goodbye.
All right, now I wanted to talk about a few things to close out our 2023 meeting series. This is open questions for 2024. So this was something that Dick Gordon, who was in this meeting, talking about some of the things about atlases. He sent me these questions, and I wanted to go over them a little bit. And if people are interested in collaborating on them, please let us know. So these are open questions. This is based on his life's work, and he'd like to pursue some of these in the latter part of his life. And these are interesting questions that come from decades of work, and it has a deep history in a lot of the things that we're doing in the group. The first question actually is, I think, something that people have wondered about, but is something maybe we're uniquely suited to answer. Or I guess it's more of a statement than a question, and that is the changing network of neighbors from the point of view of one cell in an embryo. So we do work with embryo networks, and we've seen these in meetings where we have networks of cells that are connected through some criterion. It could be distance relations, it could be signaling, it could be anatomical affinity, and all of these, relationships can form a network. And that network necessarily changes over time as you get the dynamic processes of embryogenesis and of plasticity and of function and behavior. And so what he's saying here is that we would like to look at the network of neighbors, but not just, from the top down, looking at it sort of, as an observer, but from the point of view of one of the cells in an embryo. So the idea that one of the cells in this network is an observer and is observing changing connections within its own network. So imagine you're maybe a neuron or some sort of developing cell, and you're trying to establish connections with your neighbors in the embryo. And how would you go about doing that? What would your view be from the vantage point of being a single cell? What would your view of the embryo be? And what would your view of your neighbors be? So it's kind of an interesting question. We worked on some of these in agent-based models. We've developed, in my other group, we've developed things with respect to viewpoint networks. And that kind of fits into some of the stuff on embryo networks. So that might be an interesting place to bring those two things together. So I think it's a really interesting question. And people don't generally think about networks that way. They don't think about neural networks that way. They don't think about complex networks that way. And people certainly don't think about neuronal networks or neuroscience networks that way. So I think it's an interesting question, and maybe it's something we can follow up on. So there's no seed publication for this. But for the second two questions, we actually do have some seed publications. So the first one is, can we shift diatoms, or more generally cells? Now we work with diatoms. In the seed paper, you'll see that it focuses on diatoms, but we work on other types of cells as well, and this could apply to other types of cells. Can we shift diatoms or cells from one category to another with something called a compustat? And so what is a compustat? And so if we go back to 1996, Richard Gordon published a paper called Computer-Controlled Evolution of Diatoms Designed for a Compustat. This paper is not available online. It's from a very obscure journal, Nova Hedwigia Bereft. It's some German publication. So this is something you'd have to contact us for access to. But it kind of goes through the chemostat, the idea of a chemostat. And chemostat is actually something that they use experimentally in microbiology. The chemostat allows us to select for the fastest growing mutants of a microorganism population under nutrient limitations. So this is an experimental chemostat that you would put cells in, and you could actually do these kind of selection experiments for fast growing mutants. Since morphology and motility are the major distinguishing characteristics of diatoms, I propose instead construction of a compustat. And the compustat is continuous computer vision monitoring of every cell in a diatom culture chamber. So this is where you have diatoms and you're culturing them in a culture dish. And the idea would be that you would monitor every cell with a computer vision. And this is back in 1996. So, today that's not like a huge problem necessarily. You would just take video of it and you'd be able to monitor different parameters, maybe use some sort of machine learning to extract the real-time data, and it would give you information about that. And then you could use laser microbeam culling according to program criterion. So when you identify cells that are underperforming or don't have the right properties, you can zap them with a laser microbeam, and we have a population of competent diatom cells in the culture. A compustat could provide us with the ability to determine just how fast we can drive artificial selection and evolution of diatoms when we select for morphological and or behavioral mutants. So we can create mutants in diatoms as well as in microorganisms, and we can then drive artificial selection, meaning that we're doing the selecting instead of nature. So we can do this, and we can evolve populations of diatoms or other cells in a culture. But I kind of wanted to go beyond that and think about digital evolution, where we can build like digital models of cells and things like that. So we could do this same process with digital models of cells. We could use a generative model to generate a series of cells and then select upon those cells for desired traits and then see what happens with that model. So, it's kind of an interesting area. I wasn't aware of this paper. It kind of brought it to my attention, but I think it might be a nice project to do in some way. So basically it takes that idea of the chemostat for microbiology and it brings it into the realm of diatoms, but we can also bring this into the realm of digital simulation as well. Okay, so the third question involves early development of Caecilian Zapoda. And so this is a group of organisms that I pulled up here, and these are amphibians that are eyeless. They must look like worms, but they're actually amphibians. So they have this nice morphology. It's not a model system that a lot of people work with. So it's one of these obscure model organism systems that we can learn a lot about. But people have worked with this for a long time. This is a review of the literature of the translation of Saracen and Saracen from 1889. It's on the developmental history and anatomy of the Ceylon blind burrower, Ichthyophthis glutinosus. And so this is pretty obscure. I mean, you'd have to be interested in sort of literature review and maybe the history of biology as well as early development and obscure model organisms. So this doesn't involve any biology per se. It involves a review of the literature, but it can be linked to biology as well. And so this actually is a seed paper. This is a paper that Richard Gordon wrote with Vincent Fleury. This is on coupling of growth, differentiation, and morphogenesis, an integrated approach to design and embryogenesis. So in this is a book chapter, Origins of Design in Nature, which is just kind of going over things in nature that appear to be designed or whatever. And then this kind of talks about some of these. These are some of the parts of the paper. So this just goes through a number of models where you have, it talks, this is from 1990, or this is from 2012. But this talks a lot of the mechanical and physical aspects of development that we talk about in the group now. So the abstract reads, there is increasing evidence that cellular differentiation is induced by mechanical stresses, either shear, which is dislocation of different cells, compressive stresses like gastrulation and cartilage formation, or tensile stress, which is something like plant budding. And so they kind of go through this. If differentiation is induced by components of the stress tensor, it might be possible to boil down morphogenesis to a closed biomechanical equation incorporating growth, morphogenesis, and differentiation encapsulated in a single framework. In order to progress towards such closed models or discover their inadequacies, four important questions need to be answered. First, exactly what is the mechanical nature of living material? Second, what forces drive the deformation rates in the mechanical field? Third, how cellular differentiation is spatially organized. And then fourth, how differentiation spreads. And so we have a number of things here. We have constitutive equations. We have movement, growth, or combination of the two. We have the first spots of differentiation and where they're localized. And then for the fourth thing, how differentiation spreads or migrates throughout the embryo. So we have all these different components. We have equations, we have modes of change, we have some of these organizational aspects, and then additional organizational aspects. And so they talk about a lot of this. They talk about measures. They talk about things like self-organized buckling as an example of outer plane folding. They talk about differentiation being initiated at singularities of vector fields during cell migration. That's another example. And so in all of this work, they cite this paper that I talked about using caecilians and apoda. And so you can use these very obscure model organisms to inform some of these things because they offer unique examples of some of these processes. So in the paper here, they also have specific examples that are addressed, such as gastrulation, eye formation, heart looping, and tissue buckling, which kind of incorporate this framework and are potentially explained by it. So those are our open questions for 2024. People are welcome in our group to answer any kind of question they want, but these are three that perhaps if people are interested, we could follow up on.